Bart de Taeye, Center for Drug Design and Discovery, Leuven. Co-creating new drugs based on innovative academic science.
Yes, hello. Next slide, please. I'm from CD3, and we were created in 2006 by KU Leuven Research and Development with support from the European Investment Fund. Our mission is to stimulate technology transfer and close the innovation gap by translating innovative academic science into valuable assets. So we do focus on small molecule drug discovery. In our model, we work hand in hand with academic research groups or small biotechs from the beginning of the project till the end. Next slide, please. So this basically shows you that we can help, assist, deliver all the steps that are necessary to basically bridge the so-called valley of death between the academic research and then towards the market, towards the clinic. Next slide, please. We've been successful in doing this with different academic groups. As you can see, we partnered some programs with large pharmaceutical companies and also initiated or started some small companies ourselves. Next slide, please. Next slide, please. And so this is actually a nice case example where we started working on TRPM3, which was identified by Professor Thomas Voets and Joris Vriends at the University of Leuven. They came to us, they identified TRPM3, a highly expressed ion channel on nociceptor neurons. If you activate it, you get pain. It releases a neuropeptide. Knockout mice, they have a deficit in inflammatory hyperalgesia, and they also fail to develop neuropathic pain. The knockout mouse has a very nice safety profile, actually much better than, for instance, TRPV1 or TRPM8 knockout animals. And these animals, or these targets, excuse me, have been studied by pharmaceutical companies and other smaller companies. Next slide, please. You think pain, there's no need for a new treatment. Well, actually, about more than 25 million people in the US on a daily basis live with pain. Next slide, please. This is also the case in Britain. And if you click one more time, today only 1 out of 4 to 1 out of 7 patients with pain can be helped. And in those cases, the pain only goes down with 50%. Next slide, please. So there is a high cost with pain, basically because there is a huge impact on productivity. Next slide, please. And we actually developed, with the research group at the University of Leuven, TRPM3 antagonists. We are now at a late optimization stage. These compounds have a good efficacy, work in human neurons, good selectivity, good admit tox, bioavailability is good, and they have no influence on body core temperature or heart rate, which is the case with TRPV1 blockers. Great in vivo proof of concept, and we're 1 year from preclinical candidate. You can click through the next 2 slides. That's basically some of that in vivo data, but if you're interested, I'm looking forward to discuss further. And then please click one more slide. So we're always looking for new interesting targets, biomedical insights approaches, or new molecules. And we really look forward to working with different research groups all over the world, preferentially or ideally in Europe, to develop new drugs. Thank you.